Hey everyone, and welcome back to another lecture of Physio-Psych. Today represents the first lecture that you really need to pay attention to. We're starting into the tested material. We had a nice introduction. We gave everyone time to really get their materials together, to buy their books, to get their classes sorted out, but now we're digging into it, so no more fooling around. Now for each chapter, one thing that I want to point out is that there are learning objectives. So these learning objectives are not something that we're necessarily going to focus on for every chapter, although they are a very good representation of what you should be learning. So for today's chapter on neurons and glia, I think these are very representative of the sort of things that I might ask on a quiz. For example, contrasting the locations of the central and peripheral nervous systems, describing the different structures inside of a neuron and their function. Hint, this means definitions. This is one of the few lectures where I'm going to ask you to be able to either look up in your book, because you all have that advantage, or regurgitate the structures in a neuron and what their functions are. You should also be able to differentiate the functions of supporting cells of the central and peripheral nervous system. So in this case, we're talking about glia, and we'll get to the fact that we don't necessarily want to call these supporting cells towards the end of today's lecture. And lastly, you should be able to discuss the features and importance of the blood-brain barrier. And we're going to spend just 2 or 3 slides on the blood-brain barrier, and that's about it. But it's the fourth and most important learning objective, and I can guarantee you that each of these learning objectives is fairly representative of at least one question that could make your quiz. Starting at the very most basic level, for some of you, you may or may not know this, there are 2 different parts of your nervous system. The first is the central nervous system, which includes the brain and the spinal cord, and the second is the peripheral nervous system, which is anything outside of the brain and spinal cord. And the easiest way to remember this difference is that anything that is housed within the bones of your skull or within your spinal column is part of the central nervous system. And anything that falls outside of that bony structure that protects really the most important parts of our nervous system is considered the peripheral nervous system. So we have nerves that jump outside into, of course, our skin, but also to the eyes and other places like that, that would be considered part of the peripheral nervous system, at least in many of the cases. Starting from the most basic level, our nervous system is made up of nerves, and these are the things that are responsible for communicating between different parts of the brain, or also between the brain and the body. But one thing that's really important to point out is that the nerves, as you all think of them, if you were to have, maybe some of you have seen them if you've had a major injury, or the things that you just imagine as nerves are actually entire bundles of nerves. And so what these have is a big sheath around the outside called an epineurium. It's not something you need to remember, but that's its name. And inside, there are a lot of little nerves that have pieces of connective tissue, the myelin sheath, in most cases in the peripheral nervous system, that houses the axon that actually carries the signal. So the nerves, again, as you think of them, are it's not just one big nerve carrying information, it's actually a super highway that's filled with many different smaller nerves inside of a larger nerve bundle. And if you haven't seen a picture of a nerve, sorry for any of you that get a little bit grossed out, but this is important. We're talking about physiology here. This is what a nerve would look like in a human body. So you can actually see the nerves coming out of a spinal cord, and you can imagine these are really thick, giant pieces of giant bundles, in this case, of nerves. And so they're hundreds, thousands, if not millions of different nerve fibers that are carried through some of these different kind of nerve bundles. And so one thing that you can see here is the grouping that occurs in many of these, that they're grouped together to go out to the right places, and then they spread out, of course, down into tinier and tinier parts as you get out to places like your fingers, where you have a lot of very sensitive nerve endings. Now, one of the things that I talked about in the introductory lectures is that you should start conceptualizing what the brain is doing as part of a grander functional system. And what this means is that there's some sort of input that's coming in, we have processing, and then we have output. And this can happen at a lot of different levels. So if we start to think about the entire nervous system as one of these systems, then the inputs tend to come from things like sensory neurons, where we get information about the brightness of a light, or sound waves, or how hot or cold something is, and all kinds of things like that. And then what happens in the middle, which sometimes at the level of the spinal cord can be at a single neuron, or within the brain, this could be entire series of neurons. And so in the way that you're being presented this in the Carlson book, they're using the term interneuron in a very broad way, where you could think about this interneuron as being all of the processing that happens in the brain. But in some ways, you could also, in some examples, I guess, I should say, you could think about this as being a single individual neuron. For example, in the case of reflexes, like when you have a doctor hit the front of your knee and all of a sudden your leg shoots forward. So these kinds of things are really important to conceptualize, that the processing that happens in the middle can happen at one neuron, or it can happen at hundreds of neurons. And then, as we talked about with these ABCs of psychology, in the end, what the brain does is it guides behavior. So the output of the brain as a functional system would be some sort of motor neuron that's guiding what we're doing. And then we, of course, have this system completing this loop over and over again, where our sensory neurons provide feedback, we engage in some additional processing, and then the motor neurons provide some kind of an output again. So how do the neurons signal? Well, that's something that we're going to be getting to, and that's probably one of the biggest and most important questions of Chapter 2. But in order to understand that, we really need to understand the parts of a neuron. So you can see here that the neuron has an incredible number of important parts. And if I were you, I would pay incredible attention to this diagram. I would learn to eat, breathe, and sleep this diagram, because it's one of the most important things for so many different concepts that we're going to talk about. Going through the different parts of the neuron, we have dendrites, we have the soma, we have the axon, we have the terminals or terminal buttons, and of course at both the dendrites that receive and the axons that provide output, we have a synapse. So let's talk about these now that you've just gotten oriented in a very specific way. Dendrites represent the input for a neuron, and this is where a connection from another neuron is going to be made. So the dendrites receive information from other neurons, and this is why they have this broad branching structure, is because they might receive inputs from one other neuron onto one dendrite. They might receive inputs from 100 different neurons from 100 different parts of the brain onto one dendrite. It all depends on the type of processing that they have to do. So at these different parts of the dendrite, you can see these kind of bulges on there. Dendrites have basically dendritic spines or little almost mushroom-shaped appendages on them, and they are covered in receptors. And these receptors are designed to receive chemical messages from all of these other neurons that are making contact. So the most important thing to recognize is that these are the primary input for a neuron. This is where the information comes in. Again, start thinking about this as a functional system. And we're going to be talking about a synapse, and you saw me label this in both cases, but we're sort of seeing both the start and the finish here, where dendrites end up being what we would call the post-synaptic side. Post because they're receiving a message, and so they are one part of what makes up a synapse. The soma is this giant kind of round part of the cell, and if you've had basic biology, this is what you would think of as being the cell body. So the soma has a really important job in keeping the neuron alive, because it contains all of the different organelles that we're going to be talking about over the course of this lecture, and it is responsible for passing important proteins and things down to the axon terminals. It's responsible for making decisions about how the cell processes information based on the input from all of the dendrites. And so this is kind of the job we think of for the soma, is that this is really where the processing step happens. It may be the case that if I have a certain combination of inputs or inputs of a certain strength or all kinds of things like that, then they can cause the soma to trigger an action potential. And because the soma has many, many roles that we won't even be able to touch on in this course, things like epigenetic changes, where you can, the nucleus is causing receptors to become upregulated or downregulated so that you have more or less of them, and changing the entire function of a neuron. I think the most important message to walk away with is that the soma is really where all of the different processing or changes in processing happen within a neuron. Our axon is responsible for carrying electrical signals, or the action potentials that neurons generate in order to produce and send signals over long distances. And this is going to be something that we're going to cover in the upcoming lectures more and more, is what is an action potential and what does it do. But for the sake of brevity right now, this is an electrical charge that generates at the soma and is carried down in order to help the neuron signal. And one of the things that's important about the axon for most of the nervous system is that these axons have a coating called a myelin sheath. And this myelin sheath is basically an insulator that helps messages to be able to carry over long distances. And so there are some very important principles about how this myelin sheath acts. But the job of the axon is to transmit messages across long distances, and it does it with the help of these myelin sheaths, which help to insulate that signal and regenerate that signal. So what happens in this case is that, of course, we have a message, some sort of processing that's generated at the soma, and then we use the axon and myelin sheath to carry this all the way out to the terminal. One of the things that you see here in this diagram is this really big, broad branch on this portion of the axon here, and we would call this an axon collateral. And indeed, many axons do have this sort of branching, but I would say that it's more commonly something like you see here, where an axon reaches its end in some other area of the brain, and it has many, many small branches. But there are a few, especially certain classes of neurons, that have these big bifurcations or branches that might go off into completely separate areas of the brain. So most often these happen locally, but occasionally they split off in different directions. And this would mean that there's some special function for that too. It would mean that there's a signal that needs to get to two parts of the brain at once. For example, maybe it needs to go to the motor system to create an action, and then simultaneously to some kind of a sensory system in order to give feedback on the movement that was just planned out, so that there can be a proper reaction. So all of these different pieces of the neuron, and of course the different variations on them, have special functions. And the last thing that we need to go through on our tour of the neuron are these terminal buttons. So the terminal buttons were given their name because of the shape that you can see here. You can see pretty clearly that the end of them kind of looks like a big, broad button. And the reason that this looks this way is that this is where a lot of machinery is housed, because the terminal buttons have a very, very important job, which is that we've talked about this soma getting information and making a decision about whether the neuron should fire one of these big electrical impulses. And so if that happens, the electrical impulse will travel down the axon sheath and all the way out to the terminal buttons, and it triggers the release of chemicals called neurotransmitters. So neurotransmitters are really the primary method of signaling between neurons. So to complete our loop that we've talked about a little bit, if we have this action potential that's generated, reaches the terminal button, and it releases this chemical called a neurotransmitter, there's an incredible amount of machinery required to package these chemicals and allow them to be released at the terminal button. And then they're going to be making contact with other dendrites somewhere on some other neuron that are going to have the receptors to receive these chemical messages. And then the next neuron in line starts to make these decisions about firing an action potential or not. And so what this means is that, of course, depending on how you want to consider things, the axon as well as the terminal buttons represent kind of the primary output of the neuron. So again, keeping with this theme of a functional system, we have the input at the dendrites, we have the processing at the soma, and then we have an output message carried through the axon and out to the terminal buttons. And I should say the terminal buttons are really a part of the axon. So you can consider them as a very special part, but they're a part of what we would call the axon nonetheless. And some people call these axon terminals, or you might also hear terminal buttons. So to give you a better picture of what I've been saying this entire time, when we need to form a circuit, we would have some input coming in, and this would come into the dendrites, and you can see that we have all of these different connections being made at the dendrite, where we have a chemical messenger being released by this blue axon and being received at the dendrite as an input. These are processed by the soma, which is, in a very simple term, going to be carrying out kind of an arithmetic processing, where it's going to be taking the sum of all of these inputs, and that is used to determine whether or not we trigger this big, almost gunshot that is the action potential, this very rapid electrical signal that needs to travel long distances. So there are a lot of different processes that affect this that we'll get to throughout the semester, but for now, the processing happens at the soma. And if an action potential is fired, then it's going to travel all the way down the axon out to the axon terminals or terminal buttons, where it will signal the next neuron in line, and this will carry forward, so on and so forth, all the way through the brain. And of course, these sorts of input processing and output mechanics are happening all the time as you go through your world. Neurons are constantly firing. They're constantly getting feedback about what's happening to your body, whether you're in or out of balance, if it's hot or cold, all kinds of things like that. Okay. So the cellular machinery of a neuron is one of the places where I will apologize in advance, because it's something you need to know, and there's really just not a great way to get around this. This is just basic biology. And if you've had an introduction to biology, whether in high school or here, a lot of these things are going to sound really familiar. But this is just one of those few cases throughout the entire semester where some flashcards would be a really great thing to go back to. So one of the most important pieces of a cell is the cell membrane. And the reason that the cell membrane is so important is that the cell membrane for a neuron is very different than the membrane of a lot of other cells, in that it consists of this lipid bilayer or double lipid layer. And the reason this is so important is because it acts as really an electrical and chemical barrier. And this gives the neuron its ability to carry out its functions, things like firing action potentials, because in a way, this is kind of like the positive and the negative end of a battery. And embedded within this membrane are going to be receptors that then change this charge. And sometimes they change it very rapidly, and sometimes they change it very, very slowly. But what these do is they act as a bridge between this intracellular and extracellular space. And yet, by having the membrane in between them, we are able to create a difference between the charge inside of the cell versus outside of the cell. And this difference is what gives these neurons the ability to kind of carry around a charge or potential energy at all times. Part of what supports this is the cytoskeleton. So within the cell, we have basically a matrix of proteins that you don't really see here that are kind of carried around the entirety of the cell that allow it to hold its form. So you can think of this as being the bones or the skeleton of the cell. And then, of course, another thing that helps to keep neurons and give them their shape is the cytoplasm, which is a fluid that fills the entire cell, and it allows all of the organelles and proteins and mRNA and things to move throughout the neuron freely. One incredibly important piece of the cellular machinery, and it'll probably take you till the end of the semester to appreciate just how important the job of this one single piece of machinery in the cell is, are called microtubules. And so what these are ring-like structures of proteins, and they act as a highway for the entire neuron. So something, if we were to take a look at these, maybe from a side view, right now they just look like big tubes, but what they would really be is an entire string of proteins that form a ring together, and they have some kind of important contact sites and things on them that things can bind to. And of course, what these allow the neuron to do is something called axoplasmic transport. So it's a really important term to know, but this is basically the transport of really relevant proteins and pieces of the cell, small molecules from the cell body all the way out to the axon terminals. And this is accomplished by two different proteins, kinesin, which carries things from the cell body down to the axon terminal, and then dynein, which carries them from the terminal and back. And So one of the things that the cell body does, and we're gonna see this throughout, that's incredibly important for this axoplasmic transport, is that we need to get vesicles for the release of these neurotransmitter or chemical signals all the way out to the terminals, and we need to get new neurotransmitter when it's made, new pieces of these chemicals that are released out in order to be packaged. So there's all these things that need to make their way all the way out to this terminal button. And what that means is that this kinesin has to work really fast. So it works about twice as fast as the dynein in order to be able to do this transport. So in the end, this becomes this functional railroad for the cell. And without this, the cell would basically fall apart. It wouldn't be able to do any of the jobs that we think of as being important for a neuron. Now, getting into some specific definitions that are going to be important for the entire semester, this kinesin that we just talked about is taking things and it's basically taking them in the direction that information flows through a cell. So if we had an action potential, it would start at the cell body. And when we think of something as going with the flow of information, it goes from the cell body out to the axon terminals. So things that follow this direction of travel are called anterograde. And so kinesin's job is to accomplish anterograde axoplasmic transport. The opposite of that is something that goes backwards. And so when things go in the reverse of the direction of the neuron, we're going to be talking about this with tracers and other things when we talk about methods. When they go against the direction, we call this retrograde. So this is when things are flowing in the opposite direction. And we said, as I said just last slide, this other protein called dynein is responsible for this backwards or retrograde axoplasmic transport. So what gets transported? I've mentioned a few of these things, but to just give you the quick and kind of comprehensive list, proteins are very important things that are transported throughout the cell. They go to and from the soma, so these can be transported out to the dendrites in order to give it more receptors, or there are even some receptors that fall on the axon terminal and make a bridge between the in and outside of the cell at the terminal. We might also have lipids, which can be used in some cases to maybe repair a piece of the cell membrane if there's been some sort of damage to it. But lipids are also important in making up the vesicles that hold neurotransmitter. And then there are many organelles that are needed throughout the cell. So things like mitochondria, when we need some sort of energy exchange within the cell, might be important. And lastly, we have the synaptic vesicles that I just mentioned. So we have the synaptic vesicles and of course the neurotransmitter that gets packaged into them, which are the things that get released out of this axon terminal here in order to send a chemical message. And so the neurotransmitter and vesicles would be the last things that we need this axoplasmic transport for. Hopefully all of you have had enough biology to know that the center of the cell for almost every cell in a living organism is a nucleus, and this contains two pieces. So this is where maybe your biology may or may not hold up. So we have the nucleolus and we have chromosomes. So the nucleolus is important for producing a special organelle called a ribosome, which is important for the synthesis of proteins. And then chromosomes, basically long strands of DNA that are wrapped around histones, and becomes important for things we don't cover in this course, but are incredibly important if you want to continue in neuroscience as a career and are interested in genetics for things like epigenetic changes and modification of what proteins and other things get produced and how much. But these are the kind of two key pieces that live within the nucleus that you. So within the cytoplasm, there are going to be a number of kind of free-floating organelles. The first of these would be the endoplasmic reticulum. So the endoplasmic reticulum has two separate components. The first is the rough endoplasmic reticulum. And the rough endoplasmic reticulum is important because it's going to hold on to ribosomes and also proteins that have already been produced by the ribosomes. And these will eventually be transported to where they need to be, but when they're in the rough endoplasmic reticulum, you could think of them as being in storage. And then we also have the smooth endoplasmic reticulum, which helps to produce lipids and also to segregate some of the smaller molecules that are produced until they're needed. So all of these might be, again, used for things like later on exoplasmic transport when we need to repair lipid membranes, or we need to make changes to the myelin sheath, or things of that nature. And then also we have free-floating ribosomes, and the free-floating ribosomes, as we just said in the last slide, are produced by the nucleolus, and they are needed to help produce all kinds of different proteins needed by the cell. There's some cases there are hundreds of different proteins that a cell would produce, so these ribosomes are responsible for doing that. Another thing that we have is the Golgi apparatus, and so if you think of the endoplasmic reticulum as being kind of a storage component, the Golgi apparatus is really a packaging component. So this is where we might take multiple pieces that have been produced by other parts of the cell and group them together. So we might have proteins that are made that get wrapped into some sort of a lipid membrane so that they can be released outside the cell, or you can take specific kinds of enzymes called lysosomes and package them together so that they can be taken down to the axon terminal or other places to help break down proteins or other small molecules that the neuron no longer needs. So the main job of the Golgi apparatus is sort of this packaging of specialized groups of proteins or molecules. And then mitochondria, you've probably heard since the day that you started in some sort of biology, is the powerhouse of the cell. And basically, this is what helps to take nutrients that are given to the neurons and turn them into energy through the use of adenosine triphosphate, or ATP. So one of the things that I've made certain that I point out over the course of the past few years of teaching physio is that part of the role of the nucleus is that it creates ribosomes and it holds the DNA for the cell. But in truth, there's actually a lot more going on than that. And I've seen that there's been a progression over the years of students becoming more and more familiar with this, which is great. It means we're doing our job and we're starting to teach about these different concepts. But something that, if you haven't heard of it, called epigenetics, many people started out thinking that DNA was very static and that the DNA inside of a cell stayed the same, but that ribosomes and mRNA would work together to help cells kind of determine their identity. And this becomes the difference between a neuron and a skin cell, for example. But now we know that what can happen even with cells that are long-lasting, like a neuron, is that they can undergo genetic changes throughout their lifetime. And this happens because of the interactions with the DNA, with these proteins that I mentioned called histones, where the DNA is actually wrapped around these histones in ways where there's a constant push and pull through a number of different processes like methylation or acetylation or phosphorylation, basically just change the way that these histones and the DNA interact. And they can also cause changes, we think, in how much mRNA is produced and all kinds of things like that. So this trickles down all the way to functional changes from DNA through the level of a protein. And so speaking of this, one of the things that's then really important to say is that what we have are two basic processes that are important to know about. So when a gene is able to be active, it's able to be read and converted into mRNA. And this happens with the help of an enzyme called RNA polymerase. And so you can imagine if I have changes in how available my DNA is, then I can actually have changes in this first step of transcription. So in transcription, we have the RNA polymerase making mRNA out of or from the DNA in specific copies. And then the second step here is that this mRNA, which is able to leave the nucleus, whereas the DNA can't, is then translated by our ribosomes into a polypeptide chain, also called a protein. So this is basically the way that we get functional changes in the cell. We have DNA converted to mRNA, mRNA to protein. And these proteins can be very important. They can be receptors that determine how a cell is able to process information. Do I have more receptors or less receptors, which means the signal might come in more strongly or less strongly, things like that. So one of the last things that we're going to talk about today, we've just talked about the neurons and all of their organelles, and the other part of the brain that people tend to forget about are the glial cells. And for a long time, they've been called the supporting cells of the brain. But this is a bit of a misnomer. We shouldn't be calling the glial cells supporting cells. They were originally discovered in early anatomy, and glia means glue, and people thought that this was the glue that would hold the brain together. But we now know that they have many, many, many more roles than just. So here's an image of the different types of glial cells that we're going to be talking about. So we have our Schwann cells and oligodendrocytes that are both related to each other, but one within the central, the other within the peripheral nervous system. Microglial cells, ependymal cells, and astrocytes also found within the central nervous system, at least predominantly. So different glia have entirely different roles in the brain. Some things that are common to all of them is that they act as a buffer between neurons. So this is the reason they were called the glue of the brain early on. And as a part of this, they help to support brain structure. So they have a cytoskeleton and they sit in between neurons. And so not only are they acting as an electrical and a chemical buffer between neurons, but they also help to give the brain its structure. One of the most important jobs that glial cells have is to supply nutrients to neurons. So the glial cells are a way that we get nutrients from the body into neurons in order for that to allow them to keep doing all of their various functions. And something that puts them into the category of not just being support cells is that they actually do a few things that are important for neurotransmission, which means that not only are they playing this silent support role, but they're actually playing a very active role. And one of these things is to control the supply of neurotransmitters. And they can also modify neural signaling by both controlling how many neurotransmitters exist. They can also take up neurotransmitter from the synapse. And they send out certain signals that can modify neurotransmission. So when you put all of these things together, we stop thinking of glia as being these silent partners in the brain and see them as more of an important player. So I talked about buffer, and I mentioned this already, getting a little bit ahead of myself, but they electrically insulate neurons from one another because neurons are firing action potential. You could think of this as having crossed wires or a short circuit, so it's great to have the glia in between in order to prevent all of this different crosstalk. And of course, we have lipid bilayers on neurons and things like this, but having a little bit of insulation all throughout the brain is important as well. And one of their most important jobs, and this is another reason they got to be called support cells, is because some of the earliest glia that were visualized came into surround neural injuries. They can create scars around damage in the brain. And so because there was this huge invasion of glial cells at the point when there was a brain injury, then this was some of the things that people started to see in the very earliest examples or examinations of microglia. So first, let's just talk about the glia in the central nervous system. So all of the glia we looked at before, the oligodendrocytes, microglial cells, ependymal cells, and astrocytes exist within the central nervous system, which means that leaves the one outlier here, the Schwann cells, which exist in the periphery. Astrocytes play some of the most active roles in the brain. First and foremost, they are one of the neurons providing the biggest amount of physical support, and this is because, as you see here, they tend to interface between a number of different neurons. And another thing that they do is they act as an interface between blood vessels that go through the brain and the neurons themselves. And what they do is they are able to take nutrients from within the blood vessels, and they do two things. They both convert those nutrients into energy. So they take glucose and they will either turn it into lactate to give to the neurons for energy, or they can actually hold some of it as glycogen for short-term storage. And when we talk about ingestive behaviors, we'll see that this is kind of a process that the brain goes through and the body goes through over and over again. And the other important thing that they can do as a part of their processing is that many of them make these little branches at synapses, and so they can control levels of neurotransmitters. So sometimes this is very important. for example, if we have too much of excitatory neurotransmitters like glutamate that cause neurons to fire action potentials, that can become detrimental to the neurons over time. And so the astrocytes are playing a supporting role in taking that away. But we're also starting to see that they can play an active role in doing that at times where it's very important for changing neurotransmission. Another important role for astrocytes is in helping and being part of a team of glial cells that repairs or isolates neural damage. So the astrocytes are capable of recognizing when neurons are dead. And what they do is they divide and they create a barrier between the damaged tissue and the healthy part of the brain. And what we call this is an astrocytic scar. So they both come in and help to kind of take up the dead neurons and eat them up in this process called phagocytosis, but they also block off the area and create this astrocytic scar as a wall. The primary job of oligodendrocytes is to produce myelin sheath around axons. These myelin sheaths, when they come from an oligodendrocyte, unlike the peripheral Schwann cells, they wrap around multiple different axons in an area. And if you think about what we've said about these axons, they can travel many millimeters, or even in the human brain, many inches, to make long-range connections. And so these oligodendrocytes are going to be found locally. And the myelin sheath is basically a wrapping, almost like a cast of lipid based material that they place around the axon. And these segments that are created up of these lipid layers are about 1 to 2 microns in length. And so that seems pretty small, but if you put it into relative size, probably a human neuron is between 7 and 10 microns. So 1 to 2 microns is a fairly large stretch along an axon. And yet it still takes many of these segments. And another really important thing, and we're going to see this more and more as we talk about how the axon potential is generated and propagated, is that we have these 1 to 2 micron segments, but there's also a small break in between these segments called a node of Ranvier. And the important role of the node of Ranvier, which means it's also kind of an important role of the oligodendrocytes, is to help regenerate the action potential. So there are ion channels or that are in the middle of these nodes of Ranvier that help for the action potential to kind of pick up more electrical charge. And so the oligodendrocytes have to play this very careful role of myelinating, but also leaving spaces for these nodes of Ranvier, at least in the central. One kind of cell that we're going to talk about that your book does not talk about are called ependymal cells. And so I expect you to know these. So ependymal cells have been the more recently discovered kind of glial cell. And what we know is that these cells are responsible for producing cerebrospinal fluid and then also for circulating it, because they have these little hair-like appendages on them called cilia. And so they line parts of the brain that are filled with cerebrospinal fluid, and these big holes in the brain that you will probably have seen without knowing them as kind of dark spots on an image of fMRI or something like that are called ventricles. So the role of the ependymal cells is of course keeping these filled with cerebrospinal fluid, but then also passing it through. And what we've discovered, there have been a number of different researchers working on these things, is that this flush is really important for getting all kinds of toxins and dead tissue and other things out of the brain. So, this tends to happen at night. And one of the things that we think is a failure to do this because of different blockages or problems with the ependymal cells or all kinds of things like that can actually lead to neurodegenerative diseases like Alzheimer's, for example, or dementia. So if you have a little bit of time, you could watch this video. You can go to the slides that are there, or you can I think the video is called The Nightlife of the Brain, but there are a number of different people working on this. And I think they're a very important class of cell that's going to become even more important as we move forward. So again, although your book doesn't cover it, I think I expect that all of you would know it. One of the last glial cells that we're going to talk about in the central nervous system are the microglia. And in short, these are basically the immune cells of the brain. So one of the things that they do is they drive neuroinflammation. They can also do some signaling and play a bit of an active role in changing neurotransmission and help with the astrocytes in the role of phagocytosis, or eating up dead neurons in tissue. These cells almost look like a neuron if you look at them, because they have these things that you might confuse with dendrites. But one of the properties that's kind of cool of these glial cells is when they become activated, they can actually take these branches, and the branches will become far more extensive. And this can happen during all kinds of different states. It can happen if somebody is sick, or sometimes there are changes in these structures during things like drug withdrawal. So they play a really important role and kind of helping to come in and clean up the brain. And then when things are back to normal, they will retract down to these small structures again. In the peripheral nervous system, we have Schwann cells, and they're very easy to remember because they do the same job as the oligodendrocytes. And so I like to tell people that you can think of the two that have the most ridiculous names, and they do the same thing. But then you're probably all going to say to me, they all have ridiculous names. What's an ependymol cell? In any case, with very few exceptions, almost every neuron in the peripheral nervous system is myelinated. And if you think about this, it makes a lot of sense, right? Because in the brain, maybe you have to travel a couple of millimeters or even a few inches from place to place. But there are axons that run from the tip of your toe straight up through your spinal cord and need to get up to the brain. So we have very long range axons that need to travel or need to send signals over very long distances. And that's an incredibly important job to hand off to these Schwann cells. Another very important thing in this case is that for Schwann cells, we will have one Schwann cell, and it's going to wrap around one axon. So if you remember the oligodendrocytes, they would branch off in different directions and myelinate many axons. But the Schwann cells are more of a one-to-one relationship. A second important thing that the Schwann cells do that doesn't really happen in the brain and does not happen with the oligodendrocytes is that they are able to help guide nerve regrowth. And so you've may or may not have experienced this, but if you've ever had a major surgery, then there are some cases where they'll warn you that you may have nerve damage. And this all depends on the incision, where it was, and how long of a cut that they make, and exactly how the nerves are damaged. And they know this anatomy well enough to try and avoid things when they can, but the more major the surgery, the more likely you are to cut nerves and not have them grow back. However, if you have a short distance, they can grow back. Whereas in the brain, damage kind of sticks around and we wall it off with an astrocytic scar. So it's two different, very different processes. And this is why the brain tends to be much more vulnerable. But depending on whether it's, let's say, a small mammal or a large mammal, these Schwann cells can help to guide regrowth over pretty good distances. On the order sometimes of maybe even a couple of inches, if the situation is correct. All right, and last but not least, the blood-brain barrier. And I promised at the beginning we're only going to take a couple of slides on this. So the brain is something that we need to keep protected. And so the concept here is pretty straightforward and simple. We have a lot of substances that go through the body, and we do not want them to reach the brain. For example, we have a very important electrolyte balance of all kinds of different ions like sodium and chloride that are inside of the brain. And we don't want to disrupt that just because you drink too much water or you eat too much salt. So we keep very tight control over what goes into the brain. And the way that we do this is through the blood-brain barrier. So if you look at capillaries in the rest of the body, they're fairly porous, but capillaries in the brain are basically closed off. And so what the blood-brain barrier is doing in this way is acting as a filter where it only lets certain things through. And there are a few ways that things get through. One is that we have special transporters that'll let certain things pass. And of course, the other thing that we already talked about a little bit is that we use the help of glial cells, which is why this fits into this portion of the lecture, is that astrocytes help to take certain things out of the capillaries by making kind of a suction cup connection onto the capillary itself and being able to pull nutrients, things like glucose that the brain needs, without necessarily letting the rest of things through. And of course, one of the things that many of you probably know is that if we do get blood into the brain, it can actually be very toxic in and of itself. So not only do we not want these capillaries to be porous in the brain, because of all the toxins and things that might come in on a daily basis. But if we get large amounts of blood that come into the brain because we have some sort of damage that occurs to the blood-brain barrier, we call that a stroke. So there are a lot of other things that happen, but in effect, we get a lot of blood into the brain, and these bleeds into the brain trigger all of the astrocytes and microglia to see that part of the brain is damaged, and we start phagocytosing or eating up all the neurons, and so there's a lot that happens in a very short period of time. And actually, we've gotten a whole lot better over the past 5 to 10 years about treating stroke. We can treat it over a window of a few days now, whereas we thought the window used to be only a few hours. But nonetheless, the blood-brain barrier is a very, very important aspect of keeping our brain safe from all of these different insults that could hit it at any time. However, the blood-brain barrier is kind of smart too. And there are a couple of well-known areas there where the blood-brain barrier is a little bit more permeable. Some of these we'll talk about allow us to actually sense things like the levels of fluid in the body and blood pressure, and we have some signals that pass from the periphery that do this, but we also have some sets of neurons in the brain. And one of the more famous areas kind of back in the brain stem is called the area postrema. And so this area allows toxins to come through because it actually is the area of the brain that detects toxins. And if it detects them, it triggers us to vomit. So whenever you're feeling sick and things are not going well, or whenever you eat something that doesn't sit well in your stomach, and we start to get things in the bloodstream that are not good for us, this area postrema helps to detect them and triggers that wonderful gag reflex that we all love so much to create vomiting. So with that, that's the end. We're sitting right about an hour. I probably talk a little bit faster when I'm lecturing online than when I'm lecturing in person, especially because we don't have any stop and go. So you can expect that. I'll try not to make things much longer, but just to reiterate, please go back to your housekeeping at the beginning of this and make certain that you've tackled all of those tasks. And of course, if you have any questions on this lecture, pop into office hours, say hello, whatever you feel like you need to do, and we will move on from there. All right, thanks everyone. Have a great rest of the week, and I will catch you next time.